Hello, this is the second video about the function of mitochondria. So in this video, we will discuss about the programmed cell death due to the mitochondria. So this is the cell which that contain nucleus and mitochondria as well as cytoplasm, cell membrane. So this mitochondria contain inner mitochondrial matrix and outer mitochondrial matrix and the outer and inner membrane. So in this way, the functional part for production of ATP, regulation of innate immunity we will discuss later, but in this video, calcium homeostasis. But in this video, we will discuss about the programmed cell death. So the stem cell regulation is also the part of mitochondria function. So the stem cell regulation we will not understand here, but here we will understand about the programmed cell death, how to program cell death, and programmed cell apoptosis. Apoptosis means the cell will eat itself, damage itself, or recycle the content. So in this way, the apoptosis can be extrinsic apoptosis due to the external stimulus, intrinsic apoptosis due to the internal stimulus, and regulated necrosis. Regulated necrosis is due to the injury of the cell. So let's begin to understand with the whole picture here. So this is the mitochondria containing, I am giving a cross section for the bigger picture to understand the cell apoptosis through a mitochondrial dysfunction and mitochondrial function as a apoptosis due to the mitochondria damage. So remember, mitochondria itself can divide due to the circular DNA are present in the mitochondria. So this is the cell membrane of the cell, and here is the cytosol, and here is inside is the mitochondria. Mitochondria is double membrane organelle, outer and inner, and this is the nucleus contain DNA as a genetic material. So on the other hand, here is you can see this is the bigger picture and contain receptor. Receptor is the fast receptor. This is the family of death receptor, and this is basically the tumor necrotic factor alpha is used for the death of the cell due to the extrinsic factor. This is the extrinsic factor which that is produced from the different, for example, the immune cell. So this is the basically the regulation of innate immunity due to the apoptosis of the damaged cell, so that infected cell. So here is the fast receptor bind with the tumor necrotic factor alpha, and after here is the inside is the fast associated death domain, which that is bound with the RIPK1. This is the RIPK1 is the receptor interacting protein kinase 1. This will be interact with the protein kinase 1, and in this way the caspase 8 will be bound, and this caspase 8 will be activated through a procaspase. After this activation to trigger the T bit. This T bit is the protein which that is cleavage to form a activation, and it will be bound with the B cell lymphoma 2. But let's begin to understand another things. Here is the cleaved the RIPK1, and in this way the cleaved RIPK1 will be cleaved. After the cleaved the RIPK3 also, because the RIPK3 is a homologue, and after the procaspase 3 is also activate in the form of the caspase 3, and the caspase cascade will be occur, which that will lead to cleavage. The proteolytic cleavage will lead to cause the apoptosis, and the damaged cell will be apoptosed and recycle. So that is why the damaged cell will not progress, proliferate, and divide. So it means the cancerous cell will be inhibited by this activity. So the apoptosis will be occur. So here the procaspase 9 will be activated to produce the procaspase 9, and the APF1. This APF1 is the adapter protein apoptotic peptidase associated factor 1, which that is basically also involved, and here is the DATP, which that is also involved. So the trimeric unit of the APF1, which that is combined together. So here the BCL2, B cell lymphoma 2, which the protein will be bind with the T bit. After this binding, the activation of the backs and back. This backs and back will be bind, and after this backs and back is basically is the transporter channel creation, and to activate the MOMP. The MOMP is basically contain MPT. MPT is the mitochondrial permeability transition, which that will lead to inside protein cytochrome C. Cytochrome C will be efflux, and this efflux is the black color is the cytochrome C. Will combine together with the DATP as well as the APF1. This APF1 basically combined together, you can see in this diagram, to form a apoptosome. This apoptosome which that trigger the caspase 9 activation. After this caspase 9, due to the cleavage formation of the procaspase 9 to form a caspase 9. This caspase 9 is further cleavage formation, and in this way the apoptosis will be occur. So this is the APF1 is the adapter protein apoptotic peptidase associated factor 1. So here is the tumor necrotic factor alpha is the extrinsic factor will bind with the fast receptor. This is the death receptor, contain two domain, extracellular and intracellular. So this tumor necrotic factor alpha also bind, again I am drawing and understanding the different mechanism. In this way, the ZVAD FMK will be inhibitor of the caspase 8 protein. After this inhibition, the RIPK and RIPK3, which that will be bind. After this bind, with the FAD to activate the necrosome, not apoptosome, but in this manner the necrosome. So this necrosome will bind and will bind with the MILK2. After this binding will activate binding with it will activate the PGAM5, which that will trigger the DRP1, and the DRP1 is very important, and here is the A and T. This A and T are present in the inner mitochondrial matrix will be inhibited by this, because the A and T is important for the survival, so it will be inhibited. So in this way the apoptosis will be occur. So while on the other hand, the glutamate dehydrogenase 1 enzyme, which that will trigger after this triggering formation will lead to apoptosis. So here is on the other hand, here is the DRP1. Reactive oxygen species will activate, and glutaminolysis will be activated also. So the glutaminolysis due to the glutamate dehydrogenase enzyme, and the PGM5, which that's are present here and will activate, and it is the transporter like. So on the other hand, the mitochondrial fragmentation will be occur and increase by the DRP1. So here is the nucleus, which that's contain intrinsic factor involved for it, because the intrinsic means the inside of the cell. So remember, this is the nucleus, and this is the basically the regulatory necrosis is also involved the extrinsic factor and intrinsic factor due to the injury of the cell. But here is the intrinsic apoptosis due to the DNA damage, cell membrane damage, organelle damage, and the DNA damage also produce the caspase protein transcription to translation, and the P53 and BCL2, B cell lymphomas 2, and the BAX. This all protein will lead to activate the reactive oxygen species response. After this response of the reactive oxygen species, and here is the oxidize the intermediates which that is advanced glycation and lipid hydroperoxidase peroxides, which that will all lead to trigger inhibit the ant, and when inhibit the ant, it means the apoptosis will be occur, and reactive oxygen species will trigger the apoptosis. So this was the function of the mitochondria as a cell death. So thanks for watching.